In red is an operator. You will see that it comes directly after a promoter, and it is used to regulate promoters. Promoters have a set strength, and they can't be changed except by regulatory elements like operators. So, an operator is there to bind an inhibitory protein, like so. And when the inhibitory protein is bound, it's quite sterically bulky, so it will get in the way of any transcription factors trying to bind to the promoter. So in the presence of an inhibitory protein, the promoter doesn't work. So where does this inhibitory protein come from? Well, somewhere upstream in the DNA, there will be a coding sequence for this inhibitor. And that will be expressed usually constitutively, so all the time. And that makes our inhibitory protein as seen above. So whenever the inhibitory protein is present, the promoter is blocked, no transcription occurs. But we don't just want that, do we? We want the promoter to be promoting in transcription when and only when we want it to. So if you've ever studied the lac operon, you'll be quite familiar with this. In the presence of some environmental factor, usually a ligand, which we'll here do in green. So for the lac operon, this would be allolactose, which demonstrates the presence of lactose. When the inducer binds the repressor protein, the inhibitory protein, that makes it unbind from the operator. And when the operator is unbound, then there is nothing that stops the promoter from going. Now, there's two major types of operators that you might want to use in a SynBio project. The first one is inducible by you directly. So, for this one, it will usually be IPTG inducible. Sometimes it's xylose, but usually it's IPTG. There are far more well-characterized IPTG operators than there are for xylose. And especially in E. coli, there's a whole suite of them that you can use. So IPTG is an analog of lactose. So it actually uses the lac operator. You can have whatever promoter you want, but you have to have the lac-I repressor and the lac-O operator. IPTG is better than using allolactose because IPTG doesn't get metabolized. It's got a isopropyl thiol group, which the cell just doesn't know how to handle. So it just sticks around and your cells stay induced for as long as you want them to. A benefit of IPTG is you can induce the cells precisely when you want to. So, for example, if you're doing protein purification, you might let the cells grow up to late exponential phase and then induce them. And in that way, you can let your cells grow, and then you can induce the stress that comes with the burden of protein production. But this isn't always useful in synthetic biology projects, because in SynBio projects, you often want sensing mechanisms. And one of the easiest ways to do that is with the operator. So, this is the second type you might use. Sensing. So in this case, you would need to find, probably from existing literature or existing bacteria, you would need to find an operator and inhibitor combo where the inhibitor binds to something that you're trying to sense. Now, that might be a metal ion, for example. So if it's a metal ion, it'll be precisely the same same theory as lac operon, except it will only be going in the presence of that metal ion. Another option is maybe you can find an inhibitor operator combo where the inhibitor is heat sensitive. So in hotter conditions, maybe the inhibitor denatures somewhat so that it no longer binds to the operator. So there's lots of different types of sensing you can do, and these ones you have very little control over yourself, but it allows the cell to respond to environmental conditions, which in synthetic biology projects is very core to the ethos of why we actually use biological agents. And it is because they can sense their environment and react accordingly.